Hello and welcome back to another episode of Teach Me in 10. In this episode, I spoke to Dr. Tara Crawford Parks from DNA Genotek, who discussed how first void urine samples are an innovative, non-invasive sample type of HPV screening. Tara also introduced the Colli-P device and explained its applications. Let's get started. Hi Tara, thanks for joining us today. So I think a great place to start is by defining what exactly is first void urine and how is it different from a standard urine sample?
Hi, thanks for having me today. Excited to chat with you more about first void urine and our Colli-P device. We can start with what is first void urine. First void urine is also referred to as first catch or first pass urine, and it's that first fraction of urine produced up to the first 50 milliliters when somebody voids urine. And this fraction exhibits elevated concentrations of biomarkers, whereas a standard urine sample, which could be a midstream sample, contains a lower level of these biomarkers as the initial urine flow has washed away much of the molecular debris from the bladder, urethra, and genitals. The World Health Organization considers first void urine the preferred sample type for nucleic acid amplification tests that are targeting a lot of those biomarkers that are in that first fraction. For example, many of the assays that detect chlamydia and gonorrhea or other common STIs would use first void or first catch urine to be able to have a higher concentration of those analytes. First void urine can be collected at any time throughout the day, and it can be any urination throughout the day. This portion of the urine contains urogenital epithelial cells, mucus, and potential pathogens or extracellular vesicles, as well as DNA and RNA biomarkers that accumulate in the urethra and the lower urinary tract during the time between voids. So due to that higher concentration of biomarkers that I mentioned that are available in first void urine, it has a significant potential to support research and screening applications for oncology and infectious disease. First void urine can be much more challenging to collect accurately, only using a urine cup, as the small volume of first void urine creates a higher risk of over-diluting the sample if the initial stream is missed or if the sample is over-collected. So we've developed a Colli-P device to accurately and easily collect that first void fraction of urine, and this really allows for the standardization of the collection, and we also have stabilization solutions for that first void urine fraction, which will then provide a high quality sample that has that higher concentration of those potential biomarkers.
Great. And then could you walk us through how the Colli-P device actually works?
Yeah, I would love to. So I'll do this through a short video that will show to demonstrate how the Colli-P works. So the user uncaps the tube containing the preservative and attaches it to the Colli-P collection funnel. The Colli-P is designed with flexibility for the user, allowing collection in both a standing and a seated position. The user will urinate into the device funnel, and the urine is channeled into the collection tube first until the designated volume is collected, at which point the floater will rise, redirecting the remainder of the urine stream into the toilet, and there's no need for the user to interrupt their urine stream when using the Colli-P. After collection, the user simply detaches and recaps the tube. At this point, the sample is stable at ambient temperatures and is ready for transport back to the lab. The Colli-P devices are pre-filled with preservatives, which also allow for immediate mixing and stabilization with the first void urine sample and ensure high-quality stabilized sample. These samples can be left at room temperature and can be transported through ambient temperatures without the need for cold chain transport solutions, and from there can be processed at a processing lab. Through this innovative design, we ensure that there's a standardized volumetric collection of first void urine across various Colli-P formats. So as you can see on the graph, the volumes collected for our Colli-P 4 milliliter, 10 milliliter, 20 milliliter, and 40 milliliter variants, there's very consistent volumes collected with very small error bars in the volume that were collected across a broad group of individuals, which is not the case for urine that is collected into a urine cup. As an example, on the far right of this graph, individuals were instructed to collect 20 milliliters of first void urine into a urine cup, And as you can see from the data, the range is actually quite broad that what was actually collected from these individuals.
Great. And then is it possible to stabilize first void urine after collection?
Yeah, so as I mentioned, our Colli-Pee device they come with a preservative in the tube. We have worked to develop a number of preservatives to stabilize samples immediately upon collection. And with first void urine, we have 3 distinct solutions to capture samples. So we have the Colli-Pee UAS, the Colli-Pee UCM, and our newest device, the Colli-Pee DNA and RNA kit. And each of these have been tailored to stabilize specific biomarkers within that first void fraction. For example, the Colli-Pee UAS device is best suited for capturing cfDNA, and these are valuable analytes for the liquid biopsy applications in oncology, while our Colli-Pee UCM device was designed to stabilize urinary DNA. Now, the Colli-Pee DNA and RNA product is a new and innovative solution that can preserve both urinary DNA and RNA from human and microbial sources, which really allows for an even broader range of research applications. And so all of these stabilizing solutions, UAS, UCM, and our DNA and RNA solution, have been designed to stabilize these analytes through room temperature storage and transport at ambient temperatures, which really offers flexibility in collection workflows and logistics.
Great. And I'd love to hear your perspective on how first void urine collection compares to other HPV screening methods.
First void urine is a very versatile sample type and has been used readily in the field of disease testing for areas such as STIs and urogenital cancers. And now there's a growing body of clinical evidence demonstrating its potential utility for high-risk HPV detection for cervical cancer screening. The Colli-Pee device provides a non-invasive alternative to vaginal swabs and clinician-collected cervical swabs, with emerging data across various clinical trials to support non-inferiority to the standard of care samples that are being used for HPV detection today. There's multiple scientific publications from Europe and the UK, which have compared first void urine samples collected with the Colli-Pee device to vaginal swabs and clinician-collected samples, including trials such as the Valhudis trial, the Predictors 5.1, the COCUS trial, and the CASIS study. And each of these studies have provided a robust data set to support the potential utility of the Colli-Pee device for screening of high-risk HPV. Another critical aspect of first void urine is the collection method itself. So clinical collected cervical samples are very invasive. That limits access to those who are either unable to visit a clinical site and can have lower acceptability among individuals who maybe have experienced trauma, have cultural or religious beliefs, and that don't allow for invasive samples to be collected from those populations. First void urine really offers a non-invasive solution to allow individuals to self-collect a sample, and data collected across various studies highlights the preference that people have for the Colli-Pee device compared to vaginal collected or endocervical swab collected samples. For example, the CASIS study, which was recently published in February of 2025, showed a high preference for first void urine self-collected samples, where 2/3 of the female donors preferred home-based first void urine self-sampling using the Colli-Pee device compared to a physician-taken cervical sample. And in addition to that, over 96% of those participants found that the device was very easy to use and stated that they would use it again. So in the studies previously mentioned, the researchers found that the Colli-Pee device is a viable alternative to support expanding access to people who are facing challenging barriers to access HPV screening. And we truly believe that it offers a really exciting and innovative opportunity to expand that access.
Amazing. And then how can non-invasive sampling support removing barriers and expand access in healthcare screening?
Yeah, absolutely. So non-invasive sampling methods like first void urine have growing evidence to support how they can remove barriers to screening and, as such, increase accessibility. For those who are underscreened or never screened due to social and cultural barriers, the Colli-Pee really has a strong potential to serve as a non-invasive alternative for self-collections for HPV screening. So first, it reduces physical and psychological barriers. Many standard of care sample collection methods require invasive sampling procedures, which can really be distressing for survivors of trauma, people with disabilities, or those who have cultural sensitivities. And this then encourages participation from individuals who would avoid clinical settings due to fear, stigma, or past negative experiences. So we're really able to provide a solution that meets the needs of those individuals. Next, non-invasive sampling can really improve access for underserved populations by enabling self-collection and reducing the need to travel or take time off work to go to a clinic for sample collection. And these are often common barriers for low-income or rural populations. And additionally, non-invasive self-collection really does allow for access to screening in areas where low access to health centers and with people who may not engage with traditional healthcare systems. So we're able to provide a solution through the Colli-Pee device to support some of those barriers. And then third, we really think it empowers patient autonomy and can reduce systemic biases. So non-invasive screening options give individuals greater control over their own health data and collection process. And this allows for increased comfort and privacy, especially for marginalized groups such as the LGBTQ+ individuals or those who with non-binary gender identities who are maybe less likely to go and have an invasive sample type taken at a healthcare center. And then lastly, the Colli-Pee device is really designed to preserve those samples at the point of collection, like I mentioned earlier, which ensures that sample quality and eliminates the need for cold chain shipping. So when you're using the device in a more non-clinical setting or remote area, you can still trust the quality of the sample because we've designed these novel stabilizing liquids that are in the tube. And this really expands potential for the reach of the sample collection and reduces overall costs, simplifying study workflows for better scalability and integration into future HPV programs and research.
Amazing. Thank you for taking the time to speak to us today, Tara. That was super interesting. Thanks for joining us for another episode of Teach Me in 10, which was brought to you by DNA Genotek, a subsidiary of OraSure Technologies. As a non-invasive alternative to traditional sampling, DNA Genotek offers the Colli-Pee, a self-collection device that standardizes first void urine collection with volumetric sampling. Its unique design allows immediate mixing with preservative for sample stability, and it's available in various sizes for different applications. You can learn more through the links below, and if you enjoyed this episode, don't forget to like and subscribe to catch all our latest releases.